Hi everyone, it's Miss Lopez. Welcome to Chapter 3, Enzymes. The first topic we are going to cover for our energetics unit. Now this is not all of Chapter 3, this is only the end of Chapter 3. We began Chapter 3 back when we did macromolecules. In this part, we're going to focus on 3.3 and 3.4. Proteins act as enzymes to speed up biochemical reactions, and the regulation of metabolism occurs by regulation of enzymes. We're also going to look at changes in free energy in this topic. I'm going to remind you what we learned in Chapter 3 when we did macromolecules, that factors that can disrupt interactions determine the protein structure, or what we call denaturing. And these factors include temperature, concentration of hydrogen, or the pH, high concentration of polar substances, and nonpolar substances. Living systems depend on reactions that occur spontaneously but at very slow rates. Catalysts are substances that speed up the reactions without being permanently altered. No catalyst makes a reaction occur that cannot otherwise occur. And most biological catalysts are proteins. They are known as enzymes. And there are a few out there that are RNA molecules that are called ribozymes. First, let's talk about free energy and how enzymes can change free energy in systems. In some exergonic reactions, there is an energy barrier between reactants and products. An input of energy, also known as the activation energy, will put reactants into a transition state. Let's take a look at exergonic and endergonic reactions before moving forward. I'm going to draw a very basic chart that keeps track of changes in energy over time. Now please note that these are all generic concepts. Let's start by first looking at exergonic reactions. Exergonic reactions can be thought of as similar to exothermic reactions. However, in exergonic reactions, we're not looking at changes in heat. We are looking at changes in energy. Exergonic reactions are typically catabolic, meaning that we are going to take substrates and break them down into products. And our net change in energy is going to be negative. These reactions will occur spontaneously. So let's put our substrate along the y-axis, knowing that the term energy there is just a generic amount of energy. Usually if you were interpreting this on a graph, you would actually have data values or quantitative value on that y-axis. Now, in order to take our substrate and break the bonds to create products, we're going to have to input energy, known as the activation energy. The products will have less energy than the substrate started with. So let's take a look at the free energy change. First, the dashed line represents the amount of energy that was available in the substrate. The energy that is required to break those bonds is known as the activation energy and looks like a hill that the substrate must overcome before we get to the final data point, which is your product. Let's focus on the bottom half of this graph, where we look at the difference in energy between the substrate and the product. This is known as delta G. I'm going to put in just arbitrary numbers to kind of illustrate the delta G we are dealing with. In this case, our delta G is negative. Our change in energy is negative. Let's say that the substrate had 20 units of energy. And the product, when finished with the reaction, has 7 units of energy. When we want to calculate the delta G, we take the final amount of energy, which is 7, and subtract it from the starting amount of energy, which was 20. And what we're left with is a negative value. Now let's take a look at endergonic reactions. Using the same chart that we started with exergonic reactions, we're going to compare and contrast the two. So endergonic reactions are typically anabolic. They build products up from substrates. They have positive delta Gs. And these types of reactions will not occur spontaneously because they require the input of additional energy. Okay, let's start with some substrates along our y-axis that have energy in their bonds. The products will have more energy than what the substrate started with. So we're going to place that on our graph as well. Now let's take a look at the curve. You can see how the substrate had an initial start amount of energy, and the product has an amount of energy as well that is higher than what the substrate started with. And so the activation energy for endergonic reactions is much higher than it is for exergonic reactions. So there is an input of energy into the substrate and then a release of energy into making these products or making the bonds that are forming these products. And all of this is considered your activation energy. Despite the large activation energy, you'll notice there is still a small release of energy when we make the bonds in this product. Let's give these substrate and products some example values. The substrate is, let's say, 13 units of energy. The product has, let's say, for example, 25 units of energy. When we take the difference between the product and subtract it from the starting amount, the substrate, we find that the delta G is positive. Before the substrates can be converted into the products, it has to overcome that activation energy in both endergonic and exergonic reactions. The substrate is considered a stable molecule. The products are also considered stable. However, by overcoming that activation energy, it creates a less stable intermediate, known as a transition state. So enzymes lower the activation energy. They allow more reactants to come together and react more easily. For example, a molecule of sucrose in solution may hydrolyze in about 15 days. With sucrase, the enzyme present, the same reaction occurs in 1 second. And one of the defining characteristics of enzymes is the ending here, or the suffix ase. So let's take a look again at a free energy graph over the course of time. In this particular case, I know that this is an exergonic reaction because the reactant or your substrates have more energy than the products do down here. So this delta G is considered negative. If you looked at the reaction and paid attention to that activation energy, one of the things you're going to notice is that when there's the addition of an enzyme, there is this second line that is showing a much smaller energy of activation. In other words, the hill at this point is much lower than it is in the uncatalyzed reaction. The reactants still start with the same amount of energy, and the product still stays with the same amount of energy. It is the activation energy that changes, not the delta G. In other words, not the difference in the reactants and the products. Enzymes are highly specific. Each one catalyzes only one chemical reaction. Reactants are substrates. They bind to a specific site on the enzyme called the active site. Specificity results from the exact three-dimensional shape and chemical properties of the active site. In this diagram, our substrate is sucrose, a disaccharide, which fits perfectly into the enzyme known as sucrase. Remember that in order to break polymers, we have to add water to the reaction. And the enzyme helps to make sure that everything gets put together in the correct location. What comes out of the reaction are known as products. In this case, they are monosaccharides. And the enzyme is freed up and is able to keep doing the same reaction over and over by binding more substrates at the active site. The enzyme substrate complex, which is abbreviated as ES, is held together by hydrogen bonding, electrical attraction, or temporary covalent bonding. Typically, we write the reaction like this. Enzyme plus substrate yields the enzyme substrate complex. Then the reaction continues and you're left with an enzyme and product. It's important to note that that enzyme substrate complex is unstable and that the enzyme is still in the reaction at the end and available to keep doing the same chemical reaction. The enzyme is not changed at the end of the reaction. So now we know that enzymes change the rate at which chemical reactions occur. They speed up chemical reactions by lowering that activation energy. But how does it actually accomplish this? There are three ways this can happen. One is inducing strain. So the enzymes themselves put strain on the bonds of the substrate. They're stretched, they're pulled, and it's creating that unstable transition state and allows those bonds to be broken. Another way that enzymes can speed up chemical reactions is by substrate orientation. So enzymes make sure that the substrates are brought together in just the right way or orientation so that the bonds can form. The third way that enzymes speed up chemical reactions is by adding chemical groups. R groups may also be directly involved in the reaction, but it may be that the enzymes are replacing R groups or placing R groups in the polypeptide chains. Now in your flipped, you learned about the lock and key model, where the substrate is a perfect fit for enzymes, for the enzyme active site. But there's also what is known as the induced fit model. And you can imagine the induced fit model like a baseball in a catcher's mitt. Once the baseball enters the mitt, the hand changes slightly so that the mitt better fits around the ball. Here's a diagram of an enzyme. So all of these different pieces that you see are the different R groups of the enzyme. And this entire thing is the enzyme itself with an empty active site. The substrates can then go into the empty active sites where the chemical reactions will take place. Some enzymes require ions or other molecules in order to function. Cofactors are inorganic ions that help enzymes. Coenzymes add or remove chemical groups from the substrate. They can participate in many different reactions. The difference between cofactors and coenzymes is that coenzymes are organic. Prosthetic groups, or non-amino acid groups, can also affect enzyme function in the way that they permanently bind to their enzymes. Regarding the rates of catalyzed reactions, there is usually less enzyme than substrate present, so the reaction rate levels off when the enzyme becomes saturated. A saturated situation is when all enzyme molecules are bound to substrate molecules. You can see that when you plot the enzyme reaction rate. Here is a reaction without the enzyme added. With the enzyme added, you begin to see a very quick increase in the reaction rate. This can either be measured as the amount of product that is formed or the amount of substrate that is broken down. But after a while, you see that the reaction rate begins to level off. That is because all the enzymes are currently busy and no new reactions can occur any faster. When you're dealing with this type of graph, the reaction rate of the enzyme is found by measuring the slope of the steepest part of the graph. The formula is given on your formula sheet, but they don't specify that it's the slope of the steepest part of your graph, so be on the lookout for this type of problem. The maximum rate is used to calculate enzyme efficiency, the moles of substrate converted into product per unit time, also known as turnover. It ranges anywhere from 1 to 40 million molecules per second. Enzyme catalyzed reactions are part of the metabolic pathways. The product of one reaction is a substrate for the next. Although sometimes what happens is the products can be inhibitors. In other words, they prevent the reaction from moving forward. This is known as reversible inhibition and is common in many cells. A competitive inhibitor competes with a natural substrate for the active site. A non-competitive inhibitor binds at a site distinct from the active site. This causes a change in the enzyme shape and function. So the difference between these two is whether or not the active site is free. In the case of competitive inhibitor, the active site is blocked. In a non-competitive inhibitor, what happens is the active site is still available, but because of a molecule that causes a change in the shape of the enzyme, the substrate cannot enter into the active site, even though it is technically free and available. So you can see in this diagram how the competitive inhibitor has the same shape as the active site, and the substrate that should be going in there is physically blocked. But with non-competitive inhibitor, what happens is you get another molecule binding to a site that's different from the active site. This binding causes a change of shape in the protein, as you see here, and the substrate cannot go in. This is also known as allosteric inhibition. So with allosteric inhibition or allosteric regulation, a non-substrate molecule binds to a site other than the active site. That is known as the allosteric site. The enzyme changes shape, which alters the chemical attraction or the affinity of the active site for the substrate. And allosteric regulation can activate or inactivate enzymes. Other ways that enzyme reactions can be affected, as we talked back in the macromolecule units, include pH. Acidic side chains generates hydrogen ions and become anions. Basic side chains attract hydrogen ions and become cations. In this case, that protein tertiary structure, and thus its function, is very sensitive to the concentration of hydrogen ion, or the pH, in the environment. All enzymes have an optimal pH for activity. Taking a look at this graph, we are looking at enzymes that are found in different parts of the digestive system. You can tell by looking at the pH, for example, the acidic pH 2 for the stomach, and which enzyme is active in that area. In this case, it would be pepsin. However, in the intestines and the pancreas, where the pH is much higher, in other words, more basic, you get the activation of different enzymes in those locations. Temperature will also affect your enzyme activity. Warming increases the rates of chemical reactions, but if temperature is too high, non-covalent bonds break and inactivate the enzymes. All enzymes have an optimal temperature for activity. This graph is a generic graph showing the rate of reaction for temperature, and you can see on this graph that this, at the maximum rate, that is the optimum temperature. Leading up to that, you have an increase in reaction rate as the molecules are moving faster and there are more collisions. But beyond this, you get the breaking of bonds and the enzyme is non-functional. That's it for your notes on enzymes. I hope you guys found this video useful. Thanks for watching.